All right, our next presentation is online. And Lyon is coming to us from Washington State University. And so let me give you his intro, and then hopefully we'll get the thumbs up that he's ready to roll. So Drew is professor and endowed chair of small grains extension and research weed science at Washington State University in Pullman. His extension and research program focuses on integrated weed management and dryland small grain production systems. Prior to his arrival at WSU in 2012, Drew served 22 years as the Extension Dryland Cropping Systems Specialist at the University of Nebraska Lincoln Panhandle Research and Extension Center in Scottsbluff. First of all, served, it sounds like it was like a sentence of some sort, but nonetheless, whatever. And that's a long title. Also, he received his BS in agriculture from the University of Illinois at Urbana-Champaign and his MSc and PhD in agronomy weed science from the University of Nebraska-Lincoln. Thank you so much for joining us.
All right. Well, thank you for that introduction and your ability to get through that long position description I had in there. I am the extension weed scientist here in the Pacific Northwest. I was trained as a weed scientist and then took a 22-year side trip to work on dryland cropping systems. But even in that position, I focused on weed control, and we'll talk a little bit about that later. So we're going to talk a bit about integrated weed management and dryland wheat production systems. Start off with a little bit of description of the Pacific Northwest for those of you who are unfamiliar with the area. It is quite a bit different than much of the United States and I assume much of Canada as well. And so we'll talk about that so you have a little idea about some of the data I'm presenting and how it may or may not fit your system. And then we'll talk about herbicide resistance in general and then integrated weed management strategies for controlling some problematic weeds. And even though I'm going to use examples that may not fit your system exactly, I heard kochia and wild oat. We don't have a lot of kochia here yet, but we do have a lot of Russian thistle, which is also a tumbleweed. And wild oat is making a comeback in our area as well, again, due to herbicide resistance. So oh, there we go. So this is just a slide showing the agro ecosystem I work in Eastern Washington. It goes into a little bit into Moscow. If you can see my arrow here, the Cascade Mountains are here. And just on the east side of the Cascades is basically a temperate desert. The Cascades act as a rain shadow. And so this area in here in the Columbia Basin has agriculture primarily due to irrigation out of the Columbia River. It's just very dry. We have a few areas that get 9-10 inches of annual precip, and I'm sorry I have to talk inches. Every inch is about 25 millimeters. For those who are well established in the metric system, for some reason the U.S. has decided not to join the rest of the world on that one. But anyhow, dry area here, winter wheat fallow practice. That's what this brown area is, winter wheat fallow. And you can see it kind of running through here, our drier regions, generally up to about 12 inches of annual precip. And then we run into an area where we generally have winter wheat and a spring crop and then fallow in here. And then when we get to the green, we have winter wheat, basically continuous cropping. It's usually winter wheat, spring wheat or barley, and then a pulse crop. And then there's transition zones. Some of these lighter color areas go back and forth depending on how the climate is going. So a very significant increase in rainfall here. We're talking 9-10 inches annual precip. By the time you get to Pullman, we're all the way up to 20-22 inches of annual precip. So about every 10 miles you move east, you pick up an inch of precipitation. So quite a bit of variability, rainfall and cropping system in a fairly short distance here. The other thing that is unique. And I throw this up because Sydney, Nebraska is where I did a lot of my research previously with the University of Nebraska, and then Pullman. And this is the precipitation by month in inches. And you can see in Sydney, which I imagine this is more similar to your rainfall, our big rainfall months are May, June, and July. We didn't get a lot of precipitation in the winter, late fall, early spring. Whereas Pullman is kind of the opposite. Most of our rainfall comes in January, February, November, December, and we're actually quite dry in the summer, and that limits some of the cropping systems you can work with. It does make it an excellent winter wheat production area because the rains tend to come gently. They tend to come at a cool time of the year, so there's not a lot of evaporation. A lot of this rainfall in Nebraska would come in convective storms, so it'd come fast, you get runoff. It was warm, you get a lot of evaporation. And so what took 15, 16 inches of rainfall to grow 30 bushels, here you can grow that with less than 10 inches, just because of how the rains come and when they come. So that's a difference I think you need to understand as we talk about some of these systems is it's a little bit different here in the Pacific Northwest than a lot of other areas because we're more of a Mediterranean climate with precipitation in the winter months and pretty dry in the summer. Some of our biggest weed issues are the annual grasses. Here's a picture of a winter wheat field with Italian ryegrass. That's this one growing here, sometimes referred to as annual ryegrass. And then downy brome, a lot of growers call it cheatgrass, are two of our annual grasses that are problematic. Downy brome is a winter annual. Italian ryegrass can be actually a short-lived perennial. It can also be a winter annual or a spring annual, depending on the situation. Let's talk a little bit about herbicide resistance. I always like to start my discussions of herbicide resistance with this gentleman, Charles Darwin. Some of you may have heard of him, pretty famous scientist. And I often ask my audience, what's the name of the book he published in the 1850s that a lot of today's biological sciences are based on? And you often get on origin of the species. And that is only partially correct. The title of his book was On the Origin of the Species by Means of Natural Selection or the Preservation of Favored Races in the Struggle for Life. Scientists don't like to write short titles. This is the part of the title that gets everybody into some heated discussions, but really from a scientific standpoint, it's this natural selection, this process by which species change and adapt to changes in the environment that really is the big finding here. It's something you can see happen in a jar with fruit flies in a very short period of time. You can see it with bacteria happening very quickly. There's a really neat little video from Harvard where they have this large Petri table and they put uh, the bacteria on either end where there's no I can't think of the word right now. Antibiotic. There's no antibiotic, and then as you go in, there are increasing rates, and the bacteria comes up to the first level antibiotic, stops until one individual is able to survive on the lower dose, and then it spreads quickly to the next level, sits there for a little while, then an individual is able to live on the next highest dose, and then explodes. Anyhow, it's a fun little video if you want to take a look at that. But that's natural selection is the interesting scientific evidence. If you want to argue about origin of species, you can, but natural selection is really the scientific discovery. This is a picture. I took my first year in the Pacific Northwest, one of my first trials I put out. This is a spring barley field, and that plant growing in here is mayweed chamomile. A lot of our growers call it dog fennel. I've sprayed a group 2 herbicide on this, And you can see that the population is segregating for resistance. We have some plants that have died. We have some plants that are pretty sick, and we have some plants that don't look like anything's happened to them at all. When I go back to this field later in the season, um, this plant's going to be bigger. It's going to produce more seed. And if I come back the next year, the population will look more like this and less like these. And if I keep putting on that ALS inhibitor, that group 2 herbicide, over time, pretty much most of my population will be like this. And that's how we basically drive a population from being susceptible to primarily resistant. It's kind of a case study in natural selection. We call it artificial selection because humans are applying the selection pressure, but the principle is still the same. We basically put a selective pressure on the population over time, and that population shifts. Those that are able to survive with that new pressure are the winners. The ones who can't die out. And that's basically how herbicide resistance occurs. Now, the length of time it can take varies by the cultural practices you use, the frequency of herbicide use, what the herbicide mechanism of action is. Um, it also depends on the biology of the weed species. Different species, those that tend to have a lot of biological or genetic diversity, tend to develop resistance more quickly than those that are a little less diverse in their genetic makeup. And then the frequency of resistant biotypes. Is it one plant in a million that has the genetic makeup that gives it resistance, or is it 1 in 10 million, or maybe the mutation doesn't exist at all and you never will select it. But you put all those factors together and it determines how long it takes for you to start seeing herbicide resistance occur. I think the quickest we've seen is about 4 or 5 years with the group 2 herbicides. If some of you are old enough, looking a bit like me, you might remember Glean, one of the first chlorsulfuron, one of the first group 2 herbicides used in wheat. It was a great herbicide, controlled everything in the field until it didn't. About 5 years after its introduction, we already had Russian thistle and prickly lettuce that were resistant to that mode of action. This is from Dr. Ian Heap's weedscience.org site. It's the number of unique cases of herbicide resistance around the world, starting from 1950 and going to 2020, actually goes out to 2025. But we start in 1950 because our first synthetic organic herbicide was 2,4-D. 2,4-D was developed during World War II. When World War II ended in 1945, shortly thereafter, it became commercially available, and it was our first synthetic organic herbicide. And by 1953, we already had weed species resistant to 2,4-D. So herbicide resistance has been around as long as we've had herbicide, but you can see the selection pressure or the number of resistance issues was quite a bit lower. One, herbicides weren't used quite as much. Two, they weren't nearly as effective back in the 60s, 50s, 60s, and 70s as they have been since. And so the selection pressure was much weaker. And you couldn't rely solely on herbicides to control weeds. You had to do other things in addition because the herbicides just weren't that good. Then in the late 70s and 80s, we started coming out with some really effective herbicides. People started relying on them more and more. And you can see what happens when you rely too heavily on herbicides. You basically shift the populations to those that are resistant to your herbicides. I often tell people that we've actually been cursed to live through the golden age of herbicides. We had such effective herbicides. People kind of forgot what we used to have to do to control weeds back here, relied more and more on herbicides, and basically they've driven this herbicide resistance problem into existence. If we still were introducing herbicides at the rate we were in the 80s and 90s, we still might not care too much about it, but we haven't been. The development of new herbicides has slowed down dramatically. And so when we burn through a herbicide, there's not nearly enough new things to replace the ones we burn through. This basically looks at the earlier data, but just breaks it down by different herbicide mechanisms of action. When I was a grad student in the 80s in Nebraska, a lot of my fellow graduate students were working on triazine resistance. That's these diamond-shaped blue bars here. Atrazine was a commonly used herbicide. Of course, we grew a lot of corn in Nebraska, and atrazine was cheap and effective. And because we used a lot of it, we started developing resistance to the triazines. The ALS inhibitors, the red bars, were introduced in the mid 80s, and very quickly we developed resistance to the ALS inhibitors. It's generally a single gene mutation that confers resistance. Unfortunately, the group twos have been the group that we've used the most in wheat production, and you can see how quickly it has developed here. This is the ESPS synthase inhibitors. This is glyphosate in the blue bar. Glyphosate was actually introduced in the 70s, and there were very little resistance observed for the first 20, 30 years. Monsanto, who developed the molecule, said herbicides would never develop resistance to glyphosate because it's a very complex mechanism in the plant to be overcome. But here in the mid 1990s, we start seeing it take off. And this is the exact same time that Roundup Ready soybeans, Roundup Ready corn, Roundup Ready cotton came into being. So we went from only being able to spray Roundup before we planted and after we harvested to be able to do it before, a couple, two, three times in the crop and after. And it went out on a lot more acres, and you put it out on enough acres, on enough plants, and you eventually find those individuals that have the genes that confer resistance. So we're seeing that resistance increase quite dramatically. A lot of our early resistance was target site resistance. So that's where our inhibitors, say our herbicide, binds to an enzyme at the same location where the substrate normally would bind, and it basically blocks the substrate from being able to bind, and then you don't get the amino acids produced that you wanted to produce, and so the plant basically starves. Competitive inhibition is when the herbicide binds to the same binding site as the substrate. Non-competitive inhibition is when the herbicide binds at a different spot on the enzyme, so not the same binding site as a substrate, but when it binds, it changes the shape of the binding site, and the substrate can't bind there. Unfortunately, that's fairly simple resistance, and we understand it, and basically confers resistance to uh, weeds with the same mechanism of action. But we're starting to see more non-target site resistance show up, and this is a little more problematic because we can't say um, what all herbicides will affect. So in non-target site, you might reduce the absorption of the herbicide. You might alter the translocation so that the herbicide gets sequestered in the vacuoles in the cell. You might have different metabolisms occurring. The phoenix phenomenon is where the leaf just where the droplet was applied just dies and drops off, and so the herbicide can't translocate to the rest of the plant. Um, and these mechanisms a lot of times will confer resistance across different mechanisms of action. And it's really hard to predict what all they're going to confer resistance to. So with target site, you can say, okay, I have target site resistance to the group 2 herbicides. I'll avoid using group 2 herbicides. But with non-target site resistance, um, if you have resistance to one mechanism, it could confer resistance to several other mechanisms, and it's just very difficult to figure out. So this is kind of the new challenge in herbicide resistance is this non-target site resistance that's catching on. Palmer amaranth has a lot of non-target site resistance. We're seeing this a lot in glyphosate. So we have uh, plants that build extra wax barrier on the cuticle and don't allow as much uptake. We have membrane transport where it's being transported to areas where it doesn't affect the growth of the plant. Um, we have active site mutation. This is kind of the site-specific mutation, but we also have gene duplication. Basically, the plant makes so much EPSPS that you can't get enough glyphosate in there to block all of it. So the the enzyme can keep making its substrate, making its amino acids because you just can't get enough herbicide in the plant to make up for all the EPSPS that the plant is producing. You have the sacrificial hypersensitive source. That's a picture here of a common giant ragweed plant. Uh, these leaves have been treated with the herbicide, and basically they shrivel up, die, and before the glyphosate can get transported to the other sinks in the plant. So um, we're finding all sorts of new mechanisms of resistance and creating quite the headaches for managing herbicide resistance in our weed populations. So we all know we have resistance, and how do we go about avoiding or delaying? In a lot of cases, it's just delaying herbicide resistance. Um, if you're going to be using herbicides, you're going to be putting a selection pressure on the population, and eventually, if you keep using herbicides, you'll develop resistance. So if you take Weed Science 101 here at Washington State University or pretty much anywhere else, they'll talk about integrated weed management and the different factors that go into it. You have sanitation, mechanical, biological, cultural, and chemical. You want to use as many of these as possible, but in annual cropping systems, there are a few examples of biological control. We do have some good biological control in our perennial systems, but generally, biological control requires time for the pathogen or the biological agent, insect, whatever it is, to build up in the population. And we're perturbing annual systems a little too frequently for that to be really effective. Um, so I'm going to take that one off the list. So we have four areas where we need to concentrate our integrated weed management. But what happens if you're a no-till or a zero-till or a direct seeder, as we call them here in the Pacific Northwest? You take mechanical off the list. So now, if you're going to take biological off of mechanical, you have to do a much better job with sanitation, chemical, and cultural. And what a lot of no-tillers have done is substitute chemistry for tillage. Um, Now, organic producers, they don't use the chemical, and they tend to substitute mechanical control for chemical control. But one thing the organic producers tend to do is they get pretty sophisticated with their cultural practices. They tend to have some pretty sophisticated crop rotations. Part of that is they also have to figure out how to get nitrogen into the system. So they have to rotate in legumes and other things, but that they have more complex cropping systems. A lot of no-tillers, at least early on, merely substitute chemistry for tillage, keeping their rotations pretty much the same. And because of that, and other reasons, we're starting to develop a lot of herbicide resistance, and we're not uh, seeing as many new chemistries come along. And so chemistry is getting a little more difficult, which really pushes the pressure on sanitation and cultural practices, um, especially if you're going to try to be a no-till organic producer. That's a real challenge. You got to really think about what you're going to do there. There are three principles in integrated weed management. Use agronomic practices that limit the introduction and spread of weeds. This is basically to prevent weed problems before they start. Um, this is easy to say, not so easy to do in practice, but one of the principles. The next one is help the crop compete with weeds, and that is basically help the crop choke out weeds. And the third principle is use practices that keep weeds off balance. Anything you do over and over again is going to select for the weed biotypes and weed species that do well in that system. So the idea here is don't keep doing the same thing over and over again. Don't use the same herbicide or herbicide mechanisms of action over and over again. Don't use the same crop rotation over and over again. Don't do the same tillage practices over and over again, or you will develop resistance. So those are the three principles, and we'll talk about those a little bit and give some more specific examples. So prevent weed problems before they start. Using clean seed. Here in the Pacific Northwest, growers use 95% certified seed. So they're pretty good at using that. When I was in Nebraska, that percentage was quite a bit lower. A lot of growers would grow their own seed and only buy certified seed when they switched varieties. But you really have to know where your seed's coming from so you're not bringing seeds into the system. You want to tarp grain loads going down the road. A lot of weed infestations start along roadsides because the weed seeds sift to the top of the grain truck and then blow out and get started along roadsides. and therefore we want to control weeds on field edges, try and prevent seed set by cutting or removing those weeds. If you have livestock manure that you use, make sure you compost it to kill the weed seeds, and then manage weed seeds at harvest. This last one is a concept that the Australians have kind of introduced for us. Australian Australians have been dealing with weed resistance for a long time, and they were looking for some out of the box areas. And what they decided when they looked at wheat harvest is that they were basically spreading a lot of weeds through the back of the combine. So a weed that had developed resistance, you weren't able to control it with herbicides, survived, and then you rewarded it by putting it through the combine and spreading it throughout the field. So their thought was, what can we do um, with weeds at this point to help us reduce that issue? And so they started off with narrow windrow burning, which was highly effective, but of course has some issues with fire escaping, smoke, and health problems. And so that approach has been kind of slowing down. It was at one time the most used harvest weed seed control method, but it's not as popular anymore. What's coming on in place of it are these integrated impact mill systems. They're integrated. They used to be toe-behind systems, but now they've integrated them into various combines. This is a seed terminator that's running here close to Pullman. We have a lot of ready cops coming out of Canada, a little easier to get those here. And so we're seeing a lot of uptake of this technology in the Pacific Northwest. I believe we probably have more of these running than any other part, at least of the US. Um, and the nice thing about these is they destroy a high percentage of seed that goes through the combine, and they leave the residue out in the field. So you're not having to burn it or bale it, bale the straw and haul it off. So pretty nice system, but they are expensive and they do require a bit of fuel and power to run. The other thing is that a certain biological attribute is needed for the system to work, and that attribute is that the mature seed has to be in the head at the time of harvest. If it shatters out before then, that seed's not going to go through the combine, and you're not going to destroy it through the harvest weed seed control process. Unfortunately, a lot of the weed seeds, a problem for us, seem to shatter a little more than weeds the Australians have been working with. This is Italian ryegrass seed shatter. About 2 months beforehand, we started collecting the seed. We maximized the amount of seed about 6 weeks before harvest. And at about 6 weeks, it starts declining. And around the time of harvest, we're down to only a little over 40% of the seed still in the head. So more than half of it is shattered out. Now, Michael Walsh, who's in Australia, and I've worked with Harvest Weed Seed Control, he says if it's more than 30%, you can make progress with Harvest Weed Seed Control. Of course, it's going to be better if it's 90%. It's going to happen quicker. But also, harvest weed seed control is not a silver bullet. It's one more tool in the toolbox. It should be looked at as an extra tool to use in an integrated weed management system. My uh, brief look at wild oat is that wild oat actually shatters even more than this. So you're probably down closer to that 30% at harvest time with wild oat. So wild oat may not be the best um, candidate for fast reaction. But again, you will be destroying some of the seed, and over time, the less seed you put in the viable seed you put in the seed bank, the less you have to deal with. Okay. Oh, there we go. So help choke out weeds. Um, this is you want to do everything you can to grow a good competitive crop. And your previous speaker, Tom, mentioned this. I've put out hundreds of herbicide trials in my 30 plus year career, and herbicides always look better in a good stand of a crop than they look in a poor stand. Uh, Crop competition, herbicides rely on it, and it is probably one of your best weed control practices. We've kind of gotten out of the habit of really paying a lot of attention to this as part of your weed control practice because we've had these really good herbicides. But I think we really need to get back to really looking at doing everything we can to grow a good competitive crop, getting it out of the ground as quickly as possible. A lot of competition occurs early, so the quicker you can get your crop out of the ground and growing and competing, the better. If you can narrow up your row spacing, you can get shading more quickly. You want to use high quality seed. A lot of times your larger seed gets out of the ground quicker and competes better early on. And you want to look for crop varieties that are as competitive as possible. But I really think this is a somewhat forgotten strategy that really, in my mind, is one of your best weed control strategies you have, even if you're using herbicides. And then do not allow weeds to adapt. So crop rotation. I did a lot of crop rotation work in western Nebraska, and I'm sold on the um, benefits of a good crop rotation and helping to control weeds. So you want to select crops with varied seeding dates. You don't want them all to be seed. So winter crops, earlier spring crops, later spring crops, even summer crops, different life cycles, winter crops, spring crops. If there's a way to work a perennial crop into your system, the weeds we see in perennial crops are different than the weeds we see in annual crops. The Australians often do this. They'll do annual cropping for several cycles, and then they'll plant a pasture crop for a cycle, 3 or 4 years, and they're able to drive that weed seed bank down on the weeds that are problematic in annual cropping systems by using perennial crops. We actually see that in some of our areas where we have a lot of Italian ryegrass problems, especially in our lower, wetter areas. People will plant uh, alfalfa in those areas, and then as long as you cut that alfalfa before the Italian ryegrass sets seed, no seed goes into the seed bank for several years, and then you can come back and annual crop it again. And then choose crops with very competitive abilities. As an example, one of the studies I did in western Nebraska was looking at jointed goat grass. It was a big problem in our winter wheat fallow production areas in western Nebraska. It's jointed goat grass on the left, wheat on the right. They share a genome in common, so they can hybridize. This is the hybrid here. Fortunately, most of this seed is sterile and won't produce um, a plant, but a low percentage can produce and backcross. Seed longevity for jointed goat grass. If you're only out 1 year, which is what you're out in a winter wheat fallow situation, you've only really depleted the seed bank down to about 85%. So there's still 85% of your seed viable. If you can stay out of wheat for 2 years, you drive it down closer to about 35%. And the longer, if you can stay out 3 years, you're down to less than 10% viable seed. And this is true of a lot of annual grasses, which is why a crop rotation can be quite effective. If you can stay out and prevent new weed seeds from being produced for 2, 3, 4 years, you can drive down the seed bank fairly quickly and help you out. So we did a study in western Nebraska. We had 5 treatments. We had winter wheat fallow with tillage. The red bar, the tillage was just an undercutter or a sweep plow. Winter wheat fallow without tillage, so this is chemical fallow. And then we had winter wheat double fallow. The first year of fallow was no-till with chemistry, and the second year had sweep plow, winter wheat sunflower fallow, and winter wheat proso millet fallow. So broadleaf and a grass. We had jointed goat grass and feral rye, and we ran this for 8 years. So we went through um, 2 cycles. We started in 2 different years. We ran through 2 cycles of the 3-year rotation. And at the end of those 2 cycles, you can see that we've driven by using a 3-year rotation. So staying out of winter wheat just an extra year, then we stay out in the winter wheat fallow. We can drive that those number weed seed numbers quite low. We don't eliminate it, but we get the numbers quite low. And it didn't matter whether we were planting a broadleaf summer crop, a broadleaf grass crop, or planting no crop at all. It's basically not allowing weed seeds to be produced and letting the seed bank drain down an extra year and then going through that rotation. So merely by introducing a summer crop, which in the Great Plains was fairly easy to do because we had summer precip, we could really control and reduce the amount of winter annual grassy problems we have. And I think if you go to western Nebraska now, you'll see much less winter wheat fallow than there used to be. Um, and weeds have been a big part of that drive. And then the other things you can do besides crop rotation, rotate or combine herbicides with different mechanisms of action. So again, previous speaker Tom mentioned trying to get more than one mode of action out there that it's effective on the weeds can really help with our weed control as well. What we've tended to do is throw one mechanism of action at a time at this, and when we burn that one out, then we go to the next one until we burn that one out. And it's gotten us to the position where we have fewer and fewer herbicides now that work. Whereas if we'd thrown two different effective mechanisms at them all from the start, we'd probably have a lot more herbicides still working for us. I'm getting a little short on time, but I'll just give you a quick example here. This is a study I did. We had a field with downy brome and Italian ryegrass and winter wheat. We put several different herbicides out there, triolate, which we sell here as Avidex Microactive, a granular. It's a group 8 herbicide. Pyroxasulfone as a group 2 herbicide, the active ingredient in Zidua and Anthem Flex. Metribuzin is Sencor. And then we had a couple of group 2 herbicides, Imazamox, sold as Beyond, and Paroxylem as Powerflex. The triolate went out pre-plant incorporated. We had a strip of triolate put out about 50-foot wide, and then the next 50-foot no triolate. And then we went perpendicular with our pre-emergence and post-emergence herbicides. And this is basically the take-home message. In those strips where we didn't have the Avidex, none of these treatments provided commercially acceptable Italian ryegrass control. If we applied just Avidex without any other herbicides, again, no commercially acceptable control. But with Avidex and Zidua or Zidua Sencor, we got pretty good control of Italian ryegrass. These other herbicides really aren't all that good on Italian ryegrass, but they still work better with Avidex in them than without Avidex, so a different mode of action. With downy brome, same story. Any herbicide by itself, commercially not acceptable control of downy brome. Oh, sorry, I keep clicking that. With just Avidex, again, no commercially acceptable, but if we combine Avidex with these other treatments, pretty much any of them other than Powerflex in the spring, we got fairly decent control, and our yields were better. Really quickly, I'll talk a little bit about the Italian ryegrass. We've seen a lot of spring canola coming in the Pacific Northwest. Part of it was crop price, but a big part of it was this is Roundup Ready canola, and glyphosate was still effective for controlling Italian ryegrass. So we've had a lot of spring canola come in. But I'm a little concerned that all they're doing is spraying glyphosate. And we know we have glyphosate resistant Italian ryegrass in our orchards where we've been using that technology for a while. So I think it's only a matter of time. So this rating on control of Italian ryegrass was taken on the date when we made our first early application of PowerMax, our glyphosate. So we're just looking at the pre-plant products, and you can see any treatment that has Treflan in it has 90% or better control. Here's a nice picture of it. Here's the test plots. All these plots that are green, that's a little bit of canola and a lot of Italian ryegrass. So the Treflan's doing two things. It's keeping competition, early competition down. A lot of our competition occurs when the plants are young. And when I come across here with glyphosate later, I'm spraying a lot more plants where there's no Treflan than where there is Treflan. So if herbicide resistance is a numbers game, the more plants I treat with glyphosate, the more likely I am to find that individual that has resistance and drive herbicide resistance. So I want to put that glyphosate on as few plants as possible, and that's where the pre-plant and another mode of action really helps. If we look at the end at the control, we still got great control. Yes.
5 minutes.
Okay, I will wrap it up here.
Thank you.
So the herbicides are glyphosate works very well, so growers are happy with it, but I think that the other herbicides really important. We're also seeing a bit of a yield drag with glyphosate when applied late. So do not let weeds adapt. Rotate or combine herbicides with different mechanisms of action. Rotate use of herbicide resistance, and I would say rotate tillage practices. If you're always no tilling, introduce a little tillage every once in a while. If you're always tilling, introduce a little no-till. This is a good table that shows that whether you're at low risk, you want to use more than two modes of action. You want to use cultural, mechanical, and chemical rotation. You want to keep your weed infestations low. The fewer plants you spray with herbicides, the less chance you have of developing herbicide resistance. And with that, if I have a minute or two, I'll take a question.
Thanks so much, Drew. Just as we're checking to see if there's anyone that has a question in the room, online it asks that you mentioned that wild oats are making a comeback. Which region in that map were the wild oats coming back, or are they everywhere it's coming back?
No, they don't tend to be in our winter wheat fallow areas. Wild oat is not a winter annual weed, and so winter wheat's quite competitive with it. By the time it comes up, the winter wheat's competing with it. It's coming to be more of a problem in our annual cropping and in our winter wheat spring crop fallow areas. So where we grow more spring crops, we do see it in winter wheat, particularly poorer stands of winter wheat. Um, but again, I think that's being driven axial. Panoxidin was doing a good job of controlling it, but it no longer does. Our group twos are losing efficacy, and so we're just seeing more of it come on, but not in our winter wheat fallow areas so much.
And do you happen to know if there's any mRNA work that's being done? I've heard some of that, and maybe you are familiar with some of that, maybe not. What do you
I
think?
am not. I think there is some work being done on that, but I'm not very familiar with it.
Perfect. We can leave that until our later session.
Okay.
Um, Thank you very much, Drew.
You're welcome.
I really appreciate you joining us.
I hope it
Everyone
was
around for Drew.
useful.